Assalamualaikum warahmatullahi wabarakatuh. Hi everyone. How is your day today? I hope everything is fine. Well, today we will learn about the 5th chapter in this semester, Population Genetics. First, in this chapter, there are two subtopics, 5.1 Gene Pool Concept and 5.2 Hardy-Weinberg Law. So, we go to the learning outcome of subtopic 5.1 Gene Pool Concept. At the end of this lesson, you should be able to explain population genetics, gene pool, allele frequency, and genetic equilibrium. Alright, this is the population of KMPH biology unit. Can you spot your lecturer? Actually, what is population? Population is a group of individuals that belongs to same species occupying a particular habitat that are capable of interbreeding and producing fertile offspring. What is population genetics? Population genetics is the study of genetic variability within a population and of the evolutionary force that act on it. So then, what is the definition of gene pool? Gene pool consists of all copies of every type of allele at every locus in all members of the population. In population genetics, gene pool is the complete set of unique alleles in a species or population. This diagram shows the population genetics and its relation to allele frequency. On the left is the population of 15 frogs, and on the right is the gene pool for this population. Each frog have two alleles for particular characteristics. For example, skin color. So, for the gene pool of this frog's population, there are 30 alleles. Okay, how to calculate the gene pool? For example, there are 10,000 individuals that make up a population. In this population, a particular character is controlled by a certain gene. A gene consists of two alleles. The two alleles involved are capital letter A and small letter a. Let's assume that among the 10,000 individuals, the calculation of the gene pool is 2 alleles times 10,000 individuals, then we get 20,000 alleles. Allele frequency. So, what is the meaning of allele frequency? The definition of allele frequency is the ratio of the total number of a particular allele to the total number of all the alleles in the gene pool. So, the formula is total number of a particular allele divided by total number of all the alleles in the gene pool. Actually, there are two types of allele that's symbolized by capital letter and small letter. Alright, next is genotype frequency. Genotype frequency is the ratio of the total number of individuals with a particular genotype to the total number of all the individuals in the population. So, the formula is the total number of individuals with a particular genotype divided by total number of all the individuals in the population. For your information, there are three types of genotypes. First is homozygous dominant, that's symbolized by two capital letters. Second is heterozygous, that's symbolized by one capital letter and one small letter. And last one is homozygous recessive, that's symbolized by two small letters. Remember, genotype frequency and allele frequency is usually expressed as a decimal fraction, and the sum of all genotype frequency is 1.0. Okay, how about gene equilibrium? Gene equilibrium is when a population whose allele and genotype frequency do not change from generation to generation. A population with no net change in allele or genotype frequency over time is not undergoing evolutionary change. If allele frequency change over successive generations, evolution is occurring. Next, we'll proceed to learning outcome for subtopic 5.2 Hardy-Weinberg law. At the end of this lesson, you should be able to state the Hardy-Weinberg law and explain the 5 assumptions of Hardy-Weinberg law for genetic equilibrium. Number 1 is large population size. Second is random mating. Third is no mutation. Fourth is no migration. And lastly is no natural selection. So we're back to 1908, where Hardy-Weinberg law was created. This law was created by these two figures. First is Geoffrey Hardy, an English mathematician, and second is Wilhelm Weinberg, a German physician. They were independently identified a mathematical relationship between alleles and genotypes in population. And the rest is history. Hardy-Weinberg equilibrium states that in a population that is not evolving, allele and genotype frequency will remain constant from generation to generation. When a population evolves, the allele frequency in the population will change. After we discuss about Hardy-Weinberg law and the history behind it, next we continue with the 5 assumptions of Hardy-Weinberg law. First is large population size. Say the consequence if condition does not hold. In a small population, allele frequency fluctuates by chance over time, or we call it as genetic drift. Genetic drift is a random change in allele frequency in small breeding population from one generation to the next. The second one is random mating. Say the consequence is condition does not hold. If individuals mate within a subset of the population, for example, near neighbors or close relatives or inbreeding, random mixing of gametes does not occur and genotype frequency change. Third is no mutations. Say the consequence if condition does not hold, the gene pool is modified if mutation occur or if entire genes are deleted or duplicated. The fourth is no migration. Say the consequence if condition does not hold, no migration of individuals into or out of a population through immigration or emigration. And lastly is no natural selection. Say the consequence if condition does not hold, allele frequency change when individuals with different genotypes show consistent differences in their survival or reproductive success. The definition of natural selection is a process in which organisms with certain inherited characteristics are more likely to survive and reproduce than an organism with other characteristics. And lastly, we discuss about the Hardy-Weinberg equation. First is allele frequency. And the formula for allele frequency is P plus Q equals to 1. Second is genotype frequency. The formula of genotype frequency is P squared plus 2PQ plus Q squared equals to 1. And this is the summary for the formula. P is dominant allele frequency, Q is recessive allele frequency, P squared is homozygous dominant genotype, 2PQ is heterozygous genotype, and Q squared is homozygous recessive genotype. I think that's all for today's lesson. Thank you very much. Keep calm and study biology. Assalamualaikum warahmatullahi wabarakatuh. Bye.